Our next speaker is, our next talk is COVID-19 molecular pathway activations. And the team is Eric Zhang, Zhonglan Yue, Nishant Batra, Son Dang, and Clark Xu.
Everyone, can you see the presentation?
Yep.
Okay.
Yes.
So we're Team 5 in the hackathon. We developed a data repository to enable the discovery of gene expression pathways for treating coronavirus. And this effort was a collaboration between UAB and the UW-Madison School of Medicine and Public Health. So the question we were trying to address was, we have a virus that is of interest, and we want to find ways of tackling the problem and finding different expression pathways for researching the pathogen and finding treatments. And so our approach to this was to do a bottom-up approach to data. We had a team with a biologist on it, and we manually curated data sets for researchers to be able to discover pathways that are relevant to coronavirus. And in this manual curation, we combined the literature and also the genome sequencing. So we had gene signatures and also metadata about literature that was published, and took data from PubMed on the pathway genes. And as a user of PAGER-CoV, the database is set up to allow the researcher to easily review the literature associated with a pathway and also look at the gene signatures in the pathway of interest. And we were thinking that this kind of bottom-up approach would help researchers find a direction to research therapeutic targets and other treatments from the gene sets that they're searching for. Sorry, we have some background noise. Okay, so the sources from which we're drawing the literature and the data for this repository, they come from different commonly used, I guess, repositories. So our biggest source would be PubChem. And the way we set this up was we had our biologists look at over 10 different sources. The sources are listed here on the right, and annotate the gene sets with a description of how they might be related to COVID. And then we did a web scraping. We made a CGI call to PubMed's common gateway interface and programmatically associated the gene expression with over 1,500 COVID-related pathways. So essentially, we took the genes that were listed on these pages, these are 1,500 web pages, and associated them with a specific pathway that is easier for researchers to query. So to demonstrate the utility of this approach, we built a case study relating how a researcher might search for pathways with pneumonia and with coronavirus. So Eric will show that part.
Okay, thanks, Clark. So yeah, as a case study, in this age of big data, high throughput assays, you do all this work in the lab. You come up with your experimental design relating to SARS-CoV-2, and finally you get the data analyzed. So you have your differentially expressed genes. So let's just take a case study where you have, we were originally envisioning to identify whether there were specific pathways to secondary infection of pneumonia versus COVID-19, but we didn't get that far. So what we ended up doing was, okay, can you turn off your mic, please, Samain? Okay, thank you. Whatever. So let's just say you have 672 differentially expressed genes. And we took this analyzed gene set from a publicly available data source, And so if we run these 672 genes in Reactome, what do you get? A lot of cancer-related p53 pathways. Not very useful for you know, this virus that's running throughout the world. Now, when we set up PAGER-CoV, putting in these same 672 DEGs, well, you start to get a little more curation or more organized data. But you know, this is still a work in progress. But we see some drugs popping up, and keep in mind that this is all based on your user inputted gene set. So it basically allows you to reference literature very quickly based on your experimental results or clinical results. You know, you don't have to do the experiment yourself. You can just find a publicly available analyzed data and input it, and then you just go from there. All right, next slide. Okay, so now we're doing a COVID-19 case study of Reactome versus PAGER-CoV. Okay, so this is taken from a single cell COVID-19 clinical paper from bronchoalveolar lavage fluid. And I think they were looking at CD8 positive T cells. Okay, so we have 1,516 differentially expressed genes. Putting this into Reactome, you get, again, a lot of growth signaling pathways. I mean, I guess this might be useful, but because I don't really have a background in cancer or immunology for that matter, but for me as a biologist, it's not super useful. Now let's go to PAGER-CoV, put in these 1,516 DEGs, differentially expressed genes. Now something interesting is popping up from, you can see, okay, so the user interface isn't quite there. The curation still needs to get a little better, but you get accurate identification of the cell source, CD8 positive T cells. And then you get accurate identification, I think, of a possible COVID reposition drug. I think sarilumab is from what I did from a cursor research, it's like a human monoclonal antibody used to treat rheumatoid arthritis, and it's being repositioned for COVID-19. I don't know what the clinical trials are doing, how they're doing, but you know, inflammatory response, COVID-19, yes. Okay, so, and then most interestingly, you get PAGER-CoV identification of a druggable candidate lead. So we have here, I think you can see my cursor, but maybe you can't, probably not actually. But you get, you can click on this. So next slide, you see viral NSP14 to druggable human host. Okay, so you click on this. There's two genes that are identified. Click on either one. It'll take you to the NCBI, I think, gene page, or in any case, the government database for that gene. One of them being galactosidase alpha, the other one being inosine monophosphate dehydrogenase 2. And a cursory, just, you know, using the Google, both have been looked into for as a target for anti-inflammatory effects. So overall, I think it's a pretty useful tool. You know, this is just over 2 days of a hackathon, but really opened my eyes to what one can do as a team. Okay, next slide. So we talked about what PAGER-CoV can do for you. What can you do for PAGER-CoV? Well, you can use it. So this is online. It's on the UBright site. I welcome you all to go check it out. And then on the other hand, you can,
All right, 9 minutes, so 1 more minute.
Okay. if you're on the if you're submitting on the user side, or if you're a biologist, you can help us curate. We have a submission form. It's a prototype, and you know, the more people that are interested, whether you're a clinician or a biologist, you can curate gene sets and submit them to us. We'll validate it and then put them into PAGER Cove. On the other hand, you can even submit your experimental results. If you have something COVID-19 related and you want to get it into PAGER Cove, go ahead and fill out that submission form. Next slide. All right, so here's a link to PAGER Cove. And the conclusion was that, you know, even with a limited amount of curation that we were able to do, and it's only going to get bigger, and we've, you know, there's a lot more work to do, but we found it pretty useful, and we really kind of outperformed Reactome. And yeah, there's a lot of stuff to do. You know, we gotta curate it more and implement more molecular pathway databases and the
All right,
likes.
well, we gotta have some time for questions. So what tissues are the gene expression data from? Were they stratified?
Yeah, so we have both single cell sequencing data from cell types, from single cell types, and then we have bulk RNA sequencing. So the pneumonia came from both.
Oh, colon, I don't, what tissues?
What tissue? Bronchoalveolar lavage fluid.
Okay, and user input is ranked, scored, gene sets, or just a set of symbols?
Sorry, say that again?
The user input is ranked and scored gene sets or just a set of symbols? And what's the underlying gene comparison?
It's ranked by gene sets.
How do you keep it? How do you keep it up to date? Do you have an automated refresh?
Yeah, so
No.
our CGI call will update, and you just rerun it, and it'll re-update the links to the gene pathways.